functional unit, the nephron. And just to remind you, let's just run through the different parts of the nephron again. Here is the capillary network, here supplied by an afferent arteriole and an efferent arteriole, and the peritubular capillaries have been left out of this picture again. The glomerulus here, this capillary network, is surrounded by a capsule called Bowman's capsule, and blood is filtered from here into the Bowman's capsule, from where, which is called filtrate, and the filtrate then gets modified as it passes down the tubule, first in the proximal convoluted tubule, then down into the medulla in the loop of Henle, and then back out into the distal convoluted tubule in the cortex, and then dives back into the medulla as the collecting duct, and by the time it gets to the collecting duct, the filtrate has been modified, and the end result is urine, which is passed on to the renal pelvis, which is part of the kidney, and then to the ureter. So what we want to talk about first is the filtration process, which is going on right here. Now, the blood in these capillaries here is under pressure, as all blood in circulation is, and the pressure in the capillaries here determines how much the blood is filtered across this barrier here. And that's what we're going to talk about. We're going to have a look at what the barrier looks like, have a look at the pressure that forces the blood to be filtered, and what factors are affecting this filtration rate. The filter is like chemistry. You put something in a filter funnel with a filter paper and stuff drips through, and what gets through depends on the pore size of the filter paper. And so we need to look at what the filter actually looks like in the kidney, because it's obviously very important. So here is the first of these basic processes, filtration, and that's what we're going to talk about today. So here's that diagrammatic scheme again, showing you the blood coming into the glomerulus via the afferent arteriole into the glomerulus. Here, this blood is under pressure, and the pressure forces filtration across this membrane here to form the filtrate. Now, this is not a perfect process. So, what I mean by that is that when you filter plasma, no blood cells are going across here, all we're talking about is plasma. When we filter plasma across this membrane here, first of all, stuff goes across that you really don't want to lose, because once it's gone across into the tubule, the possibility is that you pee it out and then you lose it. So the process of filtration is not perfect, so some stuff gets across that you don't want to lose. Best example of this is glucose. Glucose is small enough to get through the filter, but you don't want to be peeing out glucose. So there's a second chance, and the second chance is called tubular reabsorption. So all of the glucose goes, all of the glucose that goes across the filter here gets reabsorbed back into the blood in a second chance, and that's called tubular reabsorption. So you get a second bite at keeping stuff in the body that you want to keep. It's not only glucose, it's other stuff as well. So we'll talk about that when we talk about glucose and reabsorption. Now, the other way in which filtration is not perfect is that it doesn't actually get rid of everything you want to get rid of. So some stuff goes through the glomerular capillaries and back out the efferent arteriole, and you'd rather not have it in the blood. You really wanted to get rid of that, but it didn't happen. So if that happens, you get a second chance at that too. So as the peritubular capillaries come round, and remember these capillaries are wrapped around the nephron, you get a chance to get rid of stuff you missed in the filtration process. So that's called tubular secretion. And so the urine is the filtrate here, minus stuff which is reabsorbed, plus stuff which is secreted, and that's what's eliminated. Neat, yeah? So that's, what else is to say on here? 80% of the plasma that enters the glomerulus is not filtered and leaves through the efferent arteriole. So here, that's why you get a second chance to eliminate stuff from the blood. And 20% of the plasma that enters the glomerulus is filtered. So, okay. So we need to talk about the filtration part first, and then we'll talk about the reabsorption and the secretion part. Is that okay? This business of what the reabsorption and secretion is there for is to give you a second shot at getting rid of what you don't want and keeping what you do want. So here's some things about glomerular filtration. It forms the glomerular filtrate in the Bowman's capsule. The blood is filtered, blah, blah, blah. I'll show you that in the next picture. Substances of molecular weight less than 70,000 can pass through the filter. So that means glucose can go through, lots of small molecules can go through, but no proteins can go through. You don't want protein in the filtrate or in the urine. If you have protein in the filtrate or the urine, it means there's a problem with the filter. It's called proteinuria. Proteinuria, any word ending in urea means in the urine. Just like anemia is in the blood. So proteinuria is usually bad news. Also, of course, there's no red blood cells in the urine. If there's red blood cells in the urine, then again you've got holes in the filter, which is bad news. Blood in the urine is called hematuria, blood in the urine, which you don't get either. So, so no blood cells or protein goes through, but a large number of molecules do go through because the cutoff is about 70,000 molecular weight. So then the filtrate composition is similar to plasma. It has all the low molecular weight stuff, but it doesn't have any proteins in it. And that's going to be significant, which we'll talk about in a bit. So what's this second point all about here? Well, we need to have a look closely at the Bowman's capsule and the glomerulus. And again, remember, this is a cartoon, it's a diagram to show you the relationships of the different cell types in the filter. So here's our blood coming in. Here's Bowman's capsule here, leading on to the proximal convoluted tubule. Here's the blood coming in by the afferent arteriole into this knot of capillaries, and then the blood comes out via the efferent arteriole. So if we have a look here at one of these capillaries and the Bowman's space right here over to here, what we see is this. Here is the lumen of the glomerular capillary. Here are the cells lining the capillary. Remember, they're called endothelial cells. Here is our old friend basement membranes, the external layer of these epithelial sheets. And here is Bowman's capsule, which is, and the capillaries are covered in these cells here with these long, thin, finger-like processes called, and these cells are called podocytes. And these finger-like processes are called foot processes. So if you imagine a capillary here in Bowman's space, here's a capillary here, it's covered by another kind of cell with finger-like processes here before you get into the Bowman's capsule. So if you cut those finger-like processes across, in a cross-section, you'll see this kind of thing here, cross-section through the fingers. They're not called fingers, they're called feet. So they're called foot processes. And so here you are, you can see these podocytes here with these finger-like processes covering the capillaries. And so when stuff goes across, filtered from the blood into Bowman's space, it has to go across the wall of the capillary here, between the endothelial cells usually, across the basement membranes, and then across the space between the foot processes here, and then out into the lumen of Bowman's capsule and on into the proximal tubule. So the actual filter is this area here. It's our old friend basement membrane. Looks a bit like the respiratory membrane, the alveolo-capillary membrane, you know. The alveolo-capillary membrane, the basement membranes are a barrier to the diffusion of oxygen and carbon dioxide. Here, it's exactly the same arrangement, but the basement membrane is now the filter for the movement of water, ions, and small molecules across into the Bowman's capsule. And so if you envisage the basement membranes as being a crisscross mesh of collagen and extracellular molecules like collagen, then the size of the mesh will determine what gets through. So if you have large holes in the mesh, then stuff will get through that shouldn't get through. If the mesh gets clogged up, then stuff's not going to go through that should go through. And both of these things happen in disease. The membrane gets clogged up, and so the filtration isn't working properly. The filter gets blown full of holes, and stuff like blood cells go through which shouldn't go through. So we can talk about the abnormalities of the filter, but the filter normally has this 70,000 molecular weight cutoff for ions and small molecules to go across via this route here. Is that okay? This is not a bad picture to illustrate what is going on here. Is that okay, clear? Any questions? All right. So that's what's happening. So the function of kidneys then depends on what stuff is filtered and how much of it is filtered. In other words, the rate at which blood is filtered by the kidneys, and that's called glomerular filtration rate, GFR. So glomerular filtration rate is an important measure of kidney function, glomerular filtration rate. So what determines how quickly your blood is filtered by the kidney? Well, the amount that's filtered by the kidney in each minute is incredible. 125 mils per minute is filtered by both kidneys. So what's that? That's a cup is about, cooking, if you're cooking, and a cup is about, what, 100 mils or 150 mils? About a cup of filtrate. Am I wrong? Sorry? 250. A cup is 250, is that right? Okay. Well, we've got 150, a bit less than that. It's still a lot.
Half a cup.
Actually, I do cook a lot. That's not fair. I do cook. But I never remember these numbers, and it's all in mils anyway. Anyway, what's my point here? Thank you for that. But there's 125 mils per minute is filtrate is formed. Are you peeing out 125 mils per minute? I'm not. No. You don't pee out 125 mils a minute. What you pee out is about 1 mil per minute. You pee out about 1 mil per minute, or at least 1 mil per minute ends up in your bladder. And so 99% of this filtrate is reabsorbed. So one of the things going across, remember I said the filtration process was not perfect, one of the things going across is too much water. So you're filtering large amounts of water, 125 mils a minute. You don't want to lose that much water, my God, you'd be in big trouble. So you reabsorb it, so you get a second chance to keep that in the body. And so 99% of this water is reabsorbed by a tubular reabsorption. Now, I said 125 mils per minute, but there's a big range, depends on your physiological state of hydration or dehydration, and we'll talk about that as well. But 125 mils is a standard number which people use. So 125 mils a minute, 99% is reabsorbed, you actually produce about 1 mil per minute of urine. And this filtration rate is dependent on the pressure favoring filtration across the filter. So the actual filtration pressure is about 10 millimeters of mercury. We're going to look at that right now, because this pressure is a result of a balance of forces within the capillary, within the Bowman space. So look, I've got a couple of pictures here which show exactly the same thing. Here's the concept. Here is our capillaries here in the glomerulus, in the Bowman's capsule. First of all, favoring filtration going in this direction with this arrow, is the blood pressure in the capillaries. And that is, and these are rough figures, I'm going to show you the same picture again in the next slide, and the numbers are slightly different. It's not the actual numbers that are important here, it's the balance of forces. So in this picture, the blood pressure in the capillaries is about 55 millimeters of mercury. Okay. Now, do you remember I said that the main difference between the plasma and the filtrate is that there are no proteins in the filtrate. Proteins are too big to go through the filter. So you've got proteins in the plasma, and you've got no proteins in the filtrate. Osmotically, what's that going to do? Remember? It's going to hold back the liquid, the fluid in the blood, because the proteins are exerting an osmotic pressure. What's that osmotic pressure called? Oncotic pressure. I made that point last time. So that's why oncotic pressure is important. So the presence of proteins in the blood, but not in the filtrate, tends to hold the water in the blood by osmosis. So we have to take off a pressure off that 55, and the oncotic pressure, sometimes called the plasma colloid osmotic pressure, same thing, opposes filtration to the value of about 30 millimeters of mercury, a lot. And finally, there's another pressure opposing filtration. Try pumping up a bicycle pump last summer. The more air you put into the pump, the harder it is to force more in. In other words, the fact you've already got air in the tire makes it difficult to push more air in. And it's the same in the case of Bowman's capsule. The Bowman's capsule is already full of fluid. So you're going to try and force more fluid in. Well, there's going to be a bit of pushback there. There's going to be a bit of pushback because there's already fluid in the Bowman's capsule and you're trying to force more in. And the pushback is hydrostatic pressure. There's hydrostatic pressure in the Bowman's capsule amounting to about 15 millimeters of mercury. So if you take all of these into account, very simple equation, 55 minus 30 minus 15, and you end up with this figure of about 10 millimeters of mercury pressure favoring filtration. And the rate of filtration is proportional to this pressure. So if this pressure varies, as it can, then the filtration rate will vary, and so your kidney performance will vary. So let's just, there's another picture here. I can't remember why I've got two the same. I guess I just like it. So this is the same thing. Here is the bone space, here are the capillaries here, and here are the different forces. The arrow is proportional, size of the arrow is proportional to the pressure. So here is the glomerular capillary blood pressure. Here they've got it at 60, going this way. And that's opposed by the fluid pressure, hydrostatic pressure in the bone space, PBS, that's this one up here, opposing. Osmotic force due to proteins in the plasma, that's the oncotic or colloid osmotic pressure going this way as well. And so in this particular case, they have 60 minus 15 minus 29. How the hell they get such an exact figure as 29, I don't know. And it ends up at 16 millimeters of mercury favoring filtration. It's not the numbers that are particularly important, it's the equation. Is that okay? So you can see, as I said already, that if these numbers vary at all, then the filtration pressure is going to vary, and the amount filtered is going to vary. For example, if protein goes across, and it shouldn't, but if protein goes across, then you're going to reduce the oncotic pressure difference here. That will affect the kidney performance. So if you're losing protein in the filtrate, that's going to mess up the equation by changing the oncotic pressure difference. And that's just one example of how the filtration rate changes. So this summarizes what I've just said. The rate of filtration is proportional to the pressure. So in normal conditions, this is regulated. If the filtration pressure determines the rate of filtration, then what can potentially happen is that as your systemic blood pressure goes up and down, just think carefully about this, as your systemic blood pressure goes up and down, as it does normally, that would indicate that your kidney function will go up and down. Yeah? But that doesn't happen. Not desirable for every time your blood pressure goes up that you have to rush to the bathroom. That doesn't happen. It's regulated. My theory is that we've developed this regulation as a survival mechanism. Because if you're an animal or early man and you're being chased by a lion and your blood pressure goes up, if you stop for a pee, you get eaten. So you don't survive. So that's my theory as why we have this very careful regulation. So, in fact, when our blood pressure goes up and down, kidney function stays fairly constant. It's the old story, isn't it? Homeostasis, keeping things constant, despite changes in the external environment. And so we are able to regulate the filtration rate. And it's regulated, I'll show you this in the next few slides, by the constriction and relaxation of the arterioles going in and out, and by sympathetic nerves. As I pointed out to you just now in an example, the plasma colloid osmotic pressure is important. That was the business about losing protein. If the filter's screwed up and you start losing protein, then you muck up the oncotic pressure difference. So that will change the filtration rate. But the important thing here is to have a look, particularly at this arterial constriction issue, because it regulates the glomerular filtration rate, and it's known in kidney speak as autoregulation. So this process called autoregulation counteracts changes in arterial blood pressure. So you don't have to stop for a pee, you don't get eaten by the lion. So let's just have a look at one of my diagrams here, hand diagrams here. Here is the glomerulus here, going into the bowman's capsule. Here is the arteriole coming in. Here, if I don't do that, I'll drop it and break it again. Here is the arteriole coming in. Here is the efferent coming out. So here they are, doing their thing. What happens to the pressure in the glomerulus if I dilate the afferent arteriole? It's going to go up or is it going to go down? I dilate the afferent arteriole. What's going to happen to the blood flow? Increase. What's going to happen to the pressure in these vessels? It's going to increase. So I can increase the pressure in the glomerulus by dilating the afferent arteriole. What happens if I constrict the afferent arteriole? What's that going to do to the pressure in the glomerulus? It's going to go down because you're restricting the blood flow. Now let's look at the efferent. What happens if I dilate the efferent arteriole? It's going to allow blood to flow out much more rapidly. So what's going to happen to the pressure? It's going to go down. Similarly, if I constrict the efferent arteriole, it's going to stop blood getting out. What's that going to do to the pressure? It's going to go up. Filtration will go up. So we can control the pressure in the glomerulus by constricting and dilating the vessels on either side of it. So that's what this picture shows you. And that's really the backbone of this process called autoregulation. So look, here we go. These conditions cause a decreased glomerular filtration rate because it decreases the pressure. Here's the blood flow in, so this is the afferent arteriole, AA. If you constrict it, you restrict the blood flow, the pressure goes down, you reduce the glomerular filtration rate. Here's the other one. If you dilate the afferent arteriole, EA, then blood flow out is increased. You also reduce the glomerular pressure here. You reduce the glomerular filtration rate. Here's the opposite, increasing the glomerular filtration rate by, first of all, constricting the efferent arteriole. That will prevent blood escaping, increase the pressure in here, increase the filtration rate. With that's indicated by the thick arrow, and the filtration rate goes up. And the same thing here, by dilating the afferent arteriole, increase the blood flow, increase the pressure, increase the filtration rate here. So here is a mechanism for controlling the pressure in the glomerulus, controlling the filtration rate, controlling the amount of filtrate you produce, and ultimately controlling the amount that you pee out. That's the essence of autoregulation. Do you get that? Is that okay? Now, there are other aspects to it, which I'll add on to in a minute, but that's the difficult part to grasp. It's not that difficult, really. It has to do with dilation and constriction of the afferent and efferent arteriole going into the glomerulus. Shall I stop talking for a minute? So this is a nice picture. I was happy to find this. So this is what it says here. Despite large changes in the arterial pressure, renal blood flow and glomerular filtration rate remain relatively constant. Constant is always good in physiology. And this is due to so you've got this constriction mechanism going on and a couple of other things which I'll point out. But how do you do it? How do you do that? So what you need is a mechanism in the nephron for sensing the amount that is filtered. So if your blood pressure goes up, you start filtering too much, you're going to have a tidal wave of filtrate coming down the tube. Not a tidal wave, really. Coming down the tube, you need a downstream sensor which says, whoa, okay, we've got too much filtrate here, feedback, and cause this business to happen. And that's what you've got. You've got a feedback mechanism which senses the rate at which flow is happening. And it has to do with the structure. I know we've looked at the nephron 3 or 4 times now, but there's something about it I haven't pointed out. You might have noticed, but I didn't mention it. It has to do with an activity of the structure called the juxtaglomerular apparatus. What's juxtaglomerular going to mean? Next to the glomerulus. Next to the glomerulus. And so this structure here senses increased tubular flow of filtrate. And if you've got too much water, you've probably got a change in sodium chloride concentration. And so we're not sure which is the most important, the increased flow or the decreased sodium chloride, which causes mostly constriction of the afferent arteriole, which shuts down the glomerular filtration rate. Yeah? You get the concept before we actually have a look at what's happening here? So look, this is what I didn't mention, but it's actually on almost all of the pictures that we've looked at so far. Here. So here is the glomerulus here. Here is the filtration. Here is the filtrate being formed. It goes down the proximal convoluted tubule, down the loop of Henle, and then back into the distal convoluted tubule. But look where it goes. It goes between the afferent and efferent arteriole here. The distal tubule actually is inserted between the inflow and the outflow of the blood. That enables you to sense what's going on in the tubules. So you've got filtration rate going on here. Whoops, blood pressure goes up. You've got too much fluid going down here. So you've got increased flow, you've got decreased sodium chloride concentration. It gets to here, and this structure says, wait a minute, we've got too much fluid here. And that enables this closeness here, enables the glomerulus, the arterioles here, to constrict or dilate to correct the increased flow of fluid in the filtrate down here. So what exactly is going on? Well, look, here it is over here. Here is the Bowman's capsule. Here is a cross-section through the capillaries. Here is the afferent arteriole coming in and the efferent coming out. And here is a cross-section through the distal tubule. See, we've cut a cross-section through here. The distal tubule is between the afferent and the efferent. And look what's present in the distal tubule wall. A special group of cells here called the macula densa. Macula. Macula means a spot. Densa means dense. So it's a dense spot. So when you look at these cells in the microscope, they look different. They look darker. And so there is a special group of cells here called the macula densa, and these cells are sensitive either to flow rate or to sodium chloride concentration. And when they sense something out of whack, they produce a substance which leaves these cells and diffuses into special cells lining the walls of the afferent arteriole here. And these cells then produce another substance which can cause contraction of the afferent. Whoa, that's cool. So there's increased flow rate. These cells react. They signal via a substance to these cells. These cells get the signal and produce another substance which constricts the afferent arteriole. There may also be something going on here in the efferent. You see, this has got special cells too. But the diagram shows that there are less here than there are here. We think that most of this effect is happening in the afferent arteriole, either constricting or relaxing the afferent. But there may be something going on in the efferent as well. So most of the attention is on this region here. So let's have a closer look at this. This whole shebang here is called the juxtaglomerular apparatus. And it is what's responsible for the process called autoregulation. So here is what it does. This is basically, we've already talked about this in words. Here it is in graph form. So here is the arterial pressure here changing, And if you didn't have any autoregulation, here is urine output here. So the urine output would increase as the arterial pressure, as the pressure in the capillary increases. But this doesn't happen. This is what happens here. This is the arterial pressure here. This is the renal blood flow. And this is the glomerular filtration rate. So as the arterial pressure goes up, the renal blood flow goes up, and then it flattens out here. Can you see that? In the range of pressure 90 to 200, so where's that? Here to here, this part here. The renal blood flow remains relative, I'm reading this, the renal blood flow remains relatively constant. And this is important so that normal changes in the arterial pressure do not cause wide ranges of renal function. So this is a graphic form of what we've been talking about for the last 15 minutes. Blood pressure goes up, but over the physiological range, renal blood flow and glomerular filtration rate remain relatively constant. Yeah? Makes sense? And it occurs because renal capillary resistance change to compensate for the pressure. That's this business here. Is that okay? We're talking about the way in which we control the rate of filtrate formation. So a feedback system is responsible for this. Cells of the afferent arteriole contain a substance called renin. Come across renin? Yeah. Old favorite physiology chestnut. Renin. Renin is an enzyme which in the blood forms another, reacts with a compound called, I'm going to talk about this in a minute, I know you've already died, reacts with a compound called angiotensinogen to form angiotensin 1 and then angiotensin 2. Does this sound vaguely familiar? And angiotensin 2 is a powerful vasoconstrictor. So I'm sure you've come across this before, and I'm going to talk about it again because it's important for the kidney, and I bet that it's going to come across again in endocrinology. So this is called the renin-angiotensin theory. Physiology will love it, although it's not completely worked out, as you'll see. So the cells of these granular cells here produce renin, which is secreted into the circulation, and in the circulation, this forms angiotensin, which causes the constriction, we think. There's also other factors going on here. This particular picture doesn't show it. This one does. Here it is again. There is also a sympathetic nerve supply to the afferent arteriole, which would almost certainly play some effect. And there's something called myogenic regulation, and this occurs to almost any blood vessel, which you may have come across, I don't know when you do cardiovascular. If you stretch it, it tends to constrict. Did you come across that before? You stretch a blood vessel, it tends to reflexly constrict. It has to do with the muscle cells surrounding the vessel. And so that if your blood pressure is too high going into the glomerulus, you're forcing blood through the afferent arteriole, increasing its pressure, stretching it, and there will be a reflex constriction. So what I've said here is that the autoregulation process seems to be a combination of this business with the afferent and afferent, the nerve supply, and the myogenic constriction. Three different components. Yeah, it's the same picture as before. So what do these cells of the macular densa do? Well, that's a bit of a black box. I didn't come here to tell you that we understand all of physiology. I came here to tell you that we understand in some cases we understand the concept, but not the details. And here's one of those. Not sure how the macula densa senses flow and sodium chloride concentration. Not sure about that. And we're not sure what it is that they make which signals to these cells here to produce renin. Not sure about that. So that's what this says. Cells of the macula densa in the distal tubule can sense apparently the flow rate and the sodium chloride concentration, and they release an active substance. Don't know what it is. Bit of a black box, this. And there are a number of different possibilities. You can look it up and see what the evidence is for these things. But whatever it is, it signals to the juxtaglomerular cells in the afferent arteriole to secrete renin. And we know what that does. That causes angiotensin production, which causes this business to go on here. In the afferent and maybe the efferent. You see there are a few question marks here. Are you okay with this? Are you, is this all right? We have to be able to control the pressure in the glomerular capillaries in order to control the rate of filtration, in order to control the volume of filtrate, in order to control how much stuff you pee out. Obviously a vital thing. This seems to be done by the juxtaglomerular apparatus, which is able to constrict and dilate the vessels going in and coming out. This is done by renin, probably by renin angiotensin, by nerve supply, and by myogenic contraction when the vessels are stretched. Called renal autoregulation. It's a homeostatic mechanism. Yeah? Can I go on? So here is one of these block diagrams, which are nice sometimes. This one's pretty good. So this summarizes what I've talked about. Increased arterial blood pressure, increased driving pressure into the glomerulus. That's, the pressure in the glomerulus will go up. Increasing the glomerular capillary pressure, that will increase the GFR. That will increase the rate of fluid flow through the tubules. Stimulates the macula densa cells to release, we don't know. This causes chemicals released that induce afferent arteriolar vasoconstriction. You could put renin here if you like, because that's what this is probably. So afferent arteriolar vasoconstriction decreases the blood flow into the glomerulus, decreases the capillary pressure to normal, decreases the glomerular filtration rate. Piece of cake. Nice diagram. Summarizes everything. Are there any questions? That's got a final couple of points to make before we quit today. But done the main stuff for today. Any questions? Neat, isn't it? I mean, this is classic physiology. With the control mechanisms and homeostasis, it's all there. So, look, this is slightly different. We'll come back to the story of what happens to the filtrate next time and how it's modified by reabsorption and secretion. So this is just a slightly different but important, and it's really harking back to the cardiovascular system. Because the kidneys are responsible not only for eliminating waste stuff that you don't want by peeing it out, but it can also affect your blood pressure. So, because there are renin-producing cells in the kidney, and when this renin is released, it doesn't stay locally. I mean, it acts locally, apparently, on the afferent and efferent arterioles, but it actually is released into the whole circulation. So if you release this renin into the whole circulation, it's going to produce angiotensin in the whole circulation. If you produce angiotensin in the whole circulation, you'll get a systemic contraction of the vessels and an increased systemic blood pressure. So you've got a danger here. Okay, you've got a nice local mechanism for afferent arterial constriction, you need that. But the danger is that the renin will get out and will cause systemic changes in blood pressure, not just changes in blood pressure in the glomerulus. So you go to the doctor takes your blood pressure, too high, oh dear. 9 times out of 10, 95 times out of 10 if you like, the doctor has no idea why you have high blood pressure. What's that kind of high blood pressure called? High blood pressure is called hypertension, yes. And that kind of hypertension, where the doctor has absolutely no clue what's causing it, is called essential hypertension. 9.5 times out of 10, you go to the doctor, he finds you have high blood pressure, has no idea why, but he can give you some drugs to bring it down. The other odd time, you've got a problem with your kidneys. Very, very small minority of times, but they have to rule that out. So they have to rule out the fact that you haven't got too much renin being produced by the kidneys. And so the glomerular filtration rate can be adjusted to counteract changes in volume due to water loss. Okay, we know that this was going to affect blood pressure. The glomerular filtration rate can be adjusted to provide long-term correction responses to altered arterial blood pressure. So this is what I'm talking about here, look. So the kidneys then provide a way of controlling systemic blood pressure as well as peeing out waste. So look, here's increased water loss, for whatever reason, diarrhea, but there are other ways of reasons as well. Decreases the plasma volume. This is pure cardiovascular now. Decreases the venous pressure. Low venous pressure acts on the renal sympathetic nerves. We talked about those, which causes constriction of the renal artery, a reduction in the glomerular filtration rate, decrease in the glomerular filtration rate, and so you pee out less water, and that corrects the loss of water. So the kidneys have a mechanism for correcting blood pressure via the changes in blood volume. So reduced venous pressure. This now is again cardiovascular. Reduced venous return to the heart. Reduced atrial pressure. Reduced stroke volume. Cardiac output. Arterial blood pressure goes down. And that has a direct effect on the glomerular filtration pressure to decrease the glomerular filtration rate and decrease the amount of water that you lose. So blood pressure then can be controlled by this same mechanism that we've been talking about in the kidneys, by changing the glomerular filtration rate and changing the amount of water that you lose, you can change the arterial blood pressure. And so the final slide is this one. You can go through these two pictures at home over the weekend. That would be a good thing to do. Again, they're pretty straightforward. You put together your cardiovascular from last term with what we've talked about this morning. Here is a reduction in arterial blood pressure, the baroreceptors, sympathetic activity, vasoconstriction, reduction in the glomerular capillary blood pressure, decrease in the GFR, decrease in the amount of urine you produce. You keep fluid and water in the body, and that increases the blood pressure to provide a long-term adjustment for blood pressure. So this is the kidney loop here. There's also a cardiovascular loop here. We're having to do with cardiac output and peripheral resistance of the blood system. So here's a way that you need to try and put together the cardiovascular with the renal, because the kidneys can determine your long-term blood pressure control.